Welcome back. Today we're talking about the details of polygenic inheritance. There's 7 exceptions that that are talked about in your book, and I'll tell you which of these are emphasized on the AP test and what each of these terms mean. So, welcome back, and let's jump into our new topic. Oh, sorry, I moved it around. Okay, so our variations from classical Mendelian genetics. What is non-Mendelian genetics? Well, Mendelian genetics still provides us with the terminology and the things that we need to know in general. They help us understand, and Mendel coined the term dominant trait, non-dominant trait, and he helped us understand how things are passed on. But there's important exceptions to this. And it turns out most human traits have the number 6, a very important exception, polygenic inheritance. And in fact, of all of these variations from classical Mendelian genetics, this is the one that AP test really focuses on. They want you to know is that in real life, many traits are not controlled by one gene. This is called polygenic inheritance. This happens in humans when we look at something such as height. So there's not just one tall gene and one short gene. Most of you are not the exact height of one of your parents, or even a blend of the two. In fact, height has to do with many factors. And there's many genes on multiple chromosomes. So because of that, we have a broad range of phenotypes or physical characteristics when it comes to height. And in fact, anytime, let's see if I could find this, yeah. Anytime that we have a whole range of possibilities in phenotype, that's because of polygenic inheritance. So whether it's skin color, which is the example emphasized in your book, whether it's height, or any other trait where we see many different genes being involved, we have polygenic inheritance. Even something that seems simple, like eye color. Sometimes people say, oh, people either have brown eyes or colored eyes. That's kind of a strange statement. Of course, if you actually spend any time at all looking at people's eyes, you know that that's also polygenic. There's many shades of brown eyes, and there's many different colors of eyes. And so anytime that we have a lot of different genes involved in a trait, we're going to see a broad range of phenotype, physical characteristics, because of a result of polygenic inheritance. Definitely something you want to know for the AP exam. A couple other important examples that we'll talk about at the time that may or may not be on the AP exam are 1 through 5. So we'll talk about these quickly. Sometimes they're on the AP exam, often they're not, but they're good to know. We might have a few multiple choice type questions on your unit test on each of these. So number 1, incomplete dominance. Okay, so we talked about with Mendelian inheritance that the flowers of the pea plants were either red or white, and the red flowers dominated the white flowers. Well, that did happen in Mendel's pea plants, but that's not always the case. Sometimes you can get the pink case. So some flowers, you can actually get a blending of 2 traits. That's called incomplete dominance. And this happens sometimes also in human traits and other things where instead of having just a purely dominant and recessive trait, we can actually have a blending taking place in the homozygous case. This actually happens in some genetic diseases as well, where we can kind of act like the genetic disease is Mendelian, but in reality, there's actually 3 cases. There's the case of the completely healthy individual, who is often homozygous dominant for this disease. There's the heterozygous case. We call that person a carrier. And then there's the person who has the disease fully, and that person is homozygous recessive. And in that heterozygous case, sometimes that person has still functioning, but less functioning trait. For example, in sickle cell disease that's a recessive, that's a result of a recessive gene that affects the population. If someone's a carrier, they have different, their red blood cells are actually altered from the healthy case where they don't have that disease at all. And so that's an example of incomplete dominance. But the easiest example is to think of flowers. But I just wanted to show you that all of these examples go beyond the example case. Another example in that your book talks about is having to do with cholesterol. And again, this is a common case in this in a lot of genetic diseases. We can have normal cells, we can have those people with mild disease, but the people who are really affected are those who get the disease genotype from both parents. This can often lead to fatal or extremely problematic disease cases. Okay, the next two ones we're talking about quickly is you can have cases of multiple alleles. And the famous example of this is blood types. Blood types is a fascinating thing to study. You will probably study it if you take any sort of biology classes in college. Unfortunately, we really don't have time to get into the biology multiple alleles, other than just to say that because there's 4 main blood types, there's really 8 main blood types. There's really hundreds of blood types, but there's 4 main blood types, A, B, AB, and O. We get some really interesting genetics of that. We'll talk about that in class. But for the AP exam, just know that it is possible to have more than 2 alleles in a population. That's similar to polygenic inheritance, but a little bit different, because in this case, you can be A type, B type, AB type, which is a type of codominant, or O type. So that's 3 different alleles. Codominant is like this AB blood, where both are dominant. This is also seen in flowers. So if we cross a white flower and a red flower, end up with a flower that has both white and red streaks, as shown here. Well, not there, but is shown in your book, then that's called codominance. And the last one you can know is that one gene can affect more than one phenotype. And there's many examples of this to know. But as far as the AP exam, we wouldn't ask you about this term. You should just know that another type of non-Mendelian genetic is if one gene affects multiple traits. That happens in human genetics as well. Okay, so those are all examples of non-Mendelian genetics. We're gonna finish by talking specifically about sex-linked traits and how they are passed on. So previously we talked about Punnett squares, and I said you can just use capital T, lowercase t. But one problem we have is when we know that not all traits are found on all chromosomes. And in fact, all non-Mendelian, Mendelian genetics is based off the assumption that, I didn't mean to close that. Mendelian genetics is based off of the assumption that these traits are independent. Remember, that was the key assumption that he made. That one trait is not causing the other trait to happen. And so that's why we have all these exceptions. In real life, that's not always the case. Sometimes the traits are right next to each other and they interact, or sometimes they're linked, or sometimes they're only found in the X or the Y chromosome. So let's talk about this last example, and this will illustrate how this works, how there can be exceptions to this. So what if we have a genetic disease that's only found on the Y chromosome? Interesting, that's called a sex-linked genetic disease. So let's talk about that. Sex-linked genetic. So let's actually say it's an X-linked. It's only found on the X chromosome. because that's a more interesting example. And this happens in real life. So what's going to happen? Does that mean only females are going to have it? No, no. Remember, males also have X chromosomes. And so in fact, how this actually works is, let's look at two individuals and how they would pass out, doing kind of a modified Punnett square here. So the one parent, let's say the mother, could be a carrier for this disease. So we're going to say X prime is diseased. The mother, of course, has two X chromosomes. The father has one X chromosome and one Y chromosome. If he has the disease, he's not a carrier, has the disease in his X chromosome, what will happen in the next generation? Well, if you remember from our previous video how to do Punnett squares, we can just do it out and see. We don't have to memorize anything. So this first offspring is going to be female, XX, and have the disease. Both of them have it. But does that mean all their daughters, if they have daughters who have the disease? No. Let's look at this next one. The next possible offspring will be a daughter that is a carrier for the disease but doesn't have it. That's good. Remember, not all carriers are going to have the full disease. And they might only be mildly affected by the trait or not affected at all. And so it's a 50-50% chance in these in this genetic disease will be passed on to the next generation. 50% chance that it will be not passed on. What about the male offspring? Well, in the male offspring, the father is not passing on his X. He's always passing on his Y. So in this case, it doesn't matter what the father had at all. The only thing that's determining whether this offspring has the disease or not is which chromosome they got from the mom. And so the male offspring also ends up being 50-50. Pretty interesting. And we could do a similar Punnett square for other examples. What if the male parent is healthy and the female parent has the disease? We could do another Punnett square. I encourage you to do that one for yourself on your own. What you should find if you do a female parent for X-linked genetic disease that has the disease, and a male parent that does not have the disease or is healthy, what you'll find is that all of the male offspring will get the disease. Interesting. But the female offspring will be 50-50. Go ahead and do that Punnett square for yourself. Check it out. Yeah, and I think we'll pause there. So there was a lot. This has been a longer video, but I hope it was helpful. We talked about Mendel's laws, and we said that they don't always work out because real genetics is complicated, especially humans. We don't just have one tall gene and one short gene, or one gene that controls each of our physical traits. We have a variety of genes. That's a good thing because it allows for polygenic inheritance. But it just makes studying this a little bit more interesting and complicated. Now, there are traits that are simpler, and often diseases come from one particular mutation on one gene. So how do we track those? Well, we need to know whether they're sex links or not, whether they show incomplete dominance or complete dominance. And once we know if there's any exception to our Mendelian rules, then we can actually have a better understanding of how these diseases are passed on. We talked about the example of incomplete dominance in these processes. Okay, that's it for today. I hope that's helpful, and I'll hope you are all having a very restful break, and see you next time.